All right, I think we'll let others just slowly join if there's a few more trickling in. So welcome everyone. Really great to see so many people joining us for the second webinar in our AgVet series. This is a series of webinars that are part of an FRDC funded project called National Coordination of Access to AgVet Products in Aquaculture. So it's all about aiming to maintain and enhance the toolkit of AgVet products available to our aquaculture industries. So in Australia, any veterinary product that's supplied for use must be first permitted by the APVMA. So in our first webinar earlier in the year, I think many of you joined us for that one. So in that webinar, we heard from the APVMA about the different types of permits available, how you can apply for a permit, and how to navigate the online services portal. But this time, we're going to dig deeper into what's required for APVMA approvals, and particularly that of new and emerging chemical technologies. So my name is Jasmine Knowles. I'm an aquatic veterinarian and have been working in aquaculture for over 10 years. And I'm currently working with Seafood Industry Australia on this FRDC project. And yeah, it's a pleasure to bring you this topic. It's something that's come up quite a lot recently from industry and other stakeholders. And so hopefully it's interesting and yeah, is adding to our collective knowledge about how to improve access to these new types of products. So just as a general introduction, because aquatic animal health challenges are becoming increasingly important around the world, the management tools that we have are evolving rapidly to keep pace. So that's leading to new types of AgVet products, and they really have the potential to complement the more traditional ones that we've seen in Australia to date. And for example, I was reading a recent study about proof of concept mRNA vaccines in fish, and they're using lipid nanoparticles for delivery of the mRNA, and it was giving almost complete protection against a serious viral disease. And it's really fascinating stuff, and it's using learnings from human medicine and terrestrial animal medicine. But as we start to apply these sorts of things into aquaculture and developing new products here, it's really important that we understand how they can actually be permitted for use, so that when we find something that works, we can actually use it in the real world. So who better to take us through the ins and outs of how we can take these emerging products from a concept to a permitted tool than the APVMA themselves. So we're very lucky to have Nathan Jeffrey, the Assistant Director of Efficacy and Safety at the APVMA, joining us today to navigate through this topic. So we will be recording this session for anyone that can't attend or can't attend for the whole time, and we'll have some time for questions at the end. So we won't record the questions, so please feel free to put questions in the chat as we go along, or hold them until the end, and you can pick Nathan's brains as much as you like. So I will hand over to you, Nathan, if you want to share your screen and take it away.
Right, thanks, Jasmine. Can you see that okay?
Yep, got it, thanks.
Yeah, brilliant. Get the right screen. First up, I'd like to quickly acknowledge the traditional custodians of Australia and their continued connection to land and sea, water, environment and community. We pay our respect to the land we live and work on, their culture and their elders past and present. So I won't go over this. Jasmine just mentioned it in the introduction. The APVMA's role is as the national regulator for agricultural and veterinary chemical products. The legislation defines what the APVMA can do. You'll find our legislation up on our webpage. But I just wanted to draw your attention to 1A of the AgVet code. And just summarising this, it defines that the health and safety of human beings, animals, and the environment is the first priority. It reflects established best practices and principles for the assessment and management of risk based on science, balances regulatory effort and any burden imposed by the system of regulation, promotes community confidence in the regulation of chemical products and their constituents. So, best practice and community confidence can change over time. So what I'm presenting now, I just want to highlight, might not be the same thing required in a few years' time, or what you've presented in the past to the APVMA might not be up to the current standards. But let's get into the more interesting stuff. Jasmine was talking about mRNA. There's other various things floating around in the media and in publications. You see something of interest, you explore it more, you're talking to researchers, and you want to take it further. So what's the next step for a registration or permit to allow it in the industry? What I want to do is make a few comments first, and it's all about the planning. If you do it the right way, you can speed up the data generation, generate the right data required for the regulator, and speed up the approval process. It'll also save you money in the long run, and studies don't need to be repeated. Speak to your potential manufacturers earlier. Lab batches and research can only take you so far. The regulator requires data generated from the final formulation using the final manufacturing process. If you use research batches, these studies may need to be repeated down the track to satisfy the regulator. I've sat on both sides of the fence now as a researcher and in regulation. Researchers, they're passionate, enthusiastic about seeing something being implemented and made through to the industry, but they also often ask the wrong questions or direct their focus in the wrong way. This may generate data, but not what is required by the regulator. So let's talk about what is required and what is assessed. So the APVMA assesses the safety, efficacy, and trade of a product. We have data guidelines that provide applicants with guidance on what data and information to prepare and provide to support their application. And we break this down into chemistry, human health, residues and trade, environment, efficacy, and target animal safety. Today, I'm going to focus on biologicals, specifically vaccines, briefly touch on bacteriophage, mainly the chemistry and efficacy and safety requirements. So on those particular topics, we have specific guidelines on our website, and I'll draw your attention to guidelines for the registration of new veterinary vaccines, adjuvants in vaccines intended for food-producing animals, guidelines on demonstrating efficacy and target animal safety of veterinary products for use in farmed fish. The APVMA also recognises guidelines and pharmacopoeia monographs from trusted overseas regulators, which are your European Pharmacopoeia, British Pharmacopoeia, US Pharmacopoeia, the US 9 CFRs, VISH, and the World Organization of Animal Health International Standards. All veterinary products manufactured in Australia must be manufactured in appropriately licensed facilities. Products manufactured outside are required to comply with GMP standards that are equivalent to these. It's based on ensuring that the manufacturing process is controlled at all stages by an audited code of GMP within a quality assurance system. There's requirements for regular self-inspection of processes and procedures, inspection of suppliers and contractors, and to be subject to regular inspections by the regulatory authority. It's expected that manufacturers will embrace a culture of continuous improvement, and that manufacturing practices will be progressively updated in line with technological developments. So quality control is the part of GMP that concern with the specifications, sampling and testing, the organisation, documentation and release procedures that ensure that the necessary and relevant tests are carried out, ensuring that this material is the materials are not released for use, nor products released for sale or supply until the quality has been judged to be satisfactory. The evaluation of the regulatory approval process that the APVMA goes through should be viewed as the final step in this quality assurance process. And that's by ensuring that the system in place systems in place are appropriate to the product concern, the data on manufacturing are appropriate, in-process and finished product tests are appropriate, stability testing, safety and efficacy studies have been undertaken or reported appropriately, batch release criteria, and future batches of the product should consistently meet the standards and specifications supplied in the registration application. Thus, assuring the end user that the product should be consistently safe and with a defined shelf life and efficacy. So if we jump into vaccines, they're a class of immunobiological. They come in many forms and presentations. Most are produced from diseased agents or their antigens. Some are products from other molecules designed to immunomodulate the physical status of the recipient. Some are produced in cell cultures, embryonic eggs, live animals, or specific growth medias. Some include adjuvants, and others have used inactivating agents in the manufacturing process. But despite all these variations, the many differences outlined, vaccines are produced group lends themselves to a standard approach to risk evaluation and management when they're developed, manufactured and evaluated under that quality assurance system. Under properly applied GMP, properly conducted and interpreted safety and efficacy studies, and proper evaluation of the data supplied during the registration process. So if we jump into chemistry and manufacturing, the chemistry dossier should address the following areas: GMP, formulation containers, manufacturing process, production control and testing of starting materials, in-process controls, control tests on the final product, consistency of production and stability. We've already touched on GMP. You should supply evidence of compliance with good manufacturing practice. Also note, GMP code has a specific annex, Annex 2, for immunobiologicals and other products of biological nature. For the formulation, you should supply information about the active constituent, maximum minimum release, end of shelf life titer, adjuvants, other excipients included in the formulation. You should reference to standards where applicable, so the USP or the BP, the function of each of these constituents in the formulation, and the quantity. If there's any formulation overages for antigen stability or volume, state what they are, and if a solute diluent is supplied with the vaccine, you should provide details of its composition. You should supply the specifications of the immediate containers. Evidence should be provided that stopper will not perish during the shelf life of the product. Some rubbers and polymers react with materials such as the oils and some adjuvants. The choice of materials should take into consideration the potential for toxicity, because some materials are known to leach and or react with the product and produce substances that can be toxic to the target species. Some plastics absorb moisture and may not be suitable for vaccine use. When we look at the manufacturing process, you should provide a flowchart of the overall process showing each step from production of the active constituent to formulation of the final product. That includes details of the production of the active from the seed material through to the active ingredient or bulk antigen. It may be easier to present this as a separate flowchart and then have individual flowcharts for the production of the vaccine. And if you've got a diluent, a separate flowchart for the diluent. You should present a detailed description of each process step on that flowchart. And include details of validation data of specific manufacturing steps, including hold times, should be included, and any justification related to the production process. When we look at starting materials, that means all components used in the production of the veterinary vaccine. These should be to appropriate monographs where they exist, and if not, should be justified why you're not using a monograph if there is one available for the excipients. We'll consider references to other compendial standards on their merits. You should provide documentation from suppliers, such as certificates of analysis and or raw material specifications, and any sterilisation processes in an annex to this part of the application dossier. Starting material can be split into 3 types. Those listed in the pharmacopoeia, and you should provide the name and code of the starting material, the title of the monograph, preferably a copy of the monograph and certificates of analysis. Similar for starting materials of non-biological origin, we require that you provide the name of the materials, description of function, material specifications, including identification and purity, and any controls and tests performed on the starting material or certificates of analysis as appropriate. Third and last is starting material of biological origins, not listed in the pharmacopoeia. And it's very similar again to the previous points. Where applicable, you should provide evidence that imported biological material of animal origin used in the manufacturing are free from agents that cause transmissible spongiform encephalopathies and free from contaminants such as extraneous agents. Materials from defined and reliable sources should be used. The specifications should note the manufacturer and the origin of the raw material. And be aware, if you're importing these sources from overseas, you will require a DAF biosecurity import permit, which is separate to the APVMA. And if the product contains a genetically modified organism, you'll require a license issued by the Office of the Gene Technology Regulator. Whenever possible, veterinary vaccine production should be based on a seedlot system and on established cell banks. This is a method of creating successive batches of vaccine from the same starting material to maintain genetic stability and prevent unwanted changes. It involves a master seed lot, which is a large, well-characterized preparation of cells or viruses that is divided into smaller, stored aliquots. An aliquot from the master seed lot is then used to create a working seed lot, which is then used in the final vaccine production batches. So for each master seed organism or seed lot, you should be assigning a specific code description for identification purposes, a record of the origin, data for isolation, storage conditions, and passage history, including purification and characterization procedures and substrates used. Characterization of the microorganism should include, at a minimum, the genus and species, strain or serotype. You should provide detailed studies and tests carried out to demonstrate purity, identity, target animal safety, pathogenicity, target animal immunogenicity of the master seed. You should provide description of the method for identifying each strain by biochemical, serological, or morphological characteristics and distinguishing it as far as possible from related strains. You should also describe the method for determining the purity of the strain. You should demonstrate that the master seed is free from extraneous agents. For live veterinary vaccines, attenuated vaccines, you should provide proof of the genetic and phenotypic stability of the attenuated characteristics. The minimum maximum number of passage levels for master seeds to production levels should be specified. You should specify and appropriately document the methods, substrates used, testing and storage of seed lots and seed passages. You should also demonstrate that the characteristics of the seed material are not changed by these subcultures. You should document the conditions under which each seed lot is stored and provide a release specification of the master seed organism. For working seed organism, it's very similar. The range of passage levels used for production, controls applied, tests carried out on the working seed and storage conditions. Again, a release specification for the working seed organism. Master cell banks, again, very similar. You should provide the source of the master cell seed, treatment of the master cell seed since origin, designation, identification of the master cell, details of the master cell seed testing method and results to demonstrate sterility, freedom from extraneous agents, and freedom from specific adventitious virus contamination, proof of freedom from mycoplasma, and stability of cell lines. Just deviating from the cell bank, some production of veterinary vaccines can't use a cell bank system and are carried out in animals. In those situations, the origin, general health and immune status of the producing animal should be verified. What disease prevention protocols and disease agents, monitoring procedures and testing specifications have been used, and define pools of the source material. If using antibiotic eggs, these have their own requirements and can be found in the various pharmacopoeias. All media and solutions used in the production of the process need to be provided. The name of the media and solution and its composition, raw material specifications for all the excipients in the media should be provided, including any tests required for freedom from specific agents. A description of the method of preparation and sterilization, and where appropriate, an import permit. Details on the storage and the shelf life of the media and solutions should also be provided. Critical in-process control tests. You should describe all analytical test procedures in detail. They should all be validated. And you should provide the results of those validation studies. You should use current pharmacopoeia monographs where available and provide copies of those monographs and certificates in an annex to this part of an application dossier. For these critical tests, you should provide the title and company test code, timing and frequency, function of the test, and description and validation of the tests. These tests are critical parts of the quality assurance system embedded in GMP. They control the progression of the manufacturing process at each stage. You should only provide details of the tests that are considered critical to allow the manufacturing process to continue to the next stage. Just want to detour slightly for inactivated vaccines. Inactivation is probably the most important in-process control test. You should provide in details the assay methodology for detoxification or inactivation of the product and the limit of detection. When conducting tests for inactivation, it's essential to take into account the possibility that under conditions of manufacture, organisms may be physically protected from inactivation. If inactivation is part of the manufacturing process, you'll also require tests for residues of inactivation and detoxifying agents, demonstrating that the inactivation or detoxifying agent has been removed, neutralized, or reduced to an acceptable level. A test for residual live virus or bacteria and or detoxification is required. This test is for complete inactivation, is performed immediately after the inactivation procedure or after subsequent process steps, enhancing the sensitivity of the test, such as concentration steps. Validation of the test should focus on the level of detection of the live virus, bacteria, or toxin. A properly designed kinetics of inactivation studies should be undertaken to confirm the conditions of inactivation, including time to complete inactivation, and the actual manufacturing process. The time required for inactivation must be significantly longer than the time needed to achieve complete inactivation. This adds a margin of safety. The maximum titer of the vaccine microorganism targeted by the selected inactivation method is established based on the inactivation kinetics data. Moving on to control tests on the final product. These should also be included in batch release specifications. You should include things like identification assay of the active ingredient, potency, identification of adjuvants if present, sterility, moisture if it's required, safety when required, extraneous agents including mycoplasma, volume, overage, recoverable volumes, pH, physical characteristics. This is not an exhaustive list, and it depends on the vaccine type coming through. For each one of those tests, you should provide the title and company test code, timing and frequency of the test, the function of the test, and a description of the test with the validation data. You may provide a copy of the test procedure document as the detailed description. This is often a good option as it demonstrates how the test has been actually conducted. The detailed description should contain sufficient information to enable us to assess the adequacy of the test method and, if applicable, whether it's consistent with a cited monograph. The batch release expiry specifications should include the name of each test, the company test code, and the limits of acceptance for results. And again, if applicable, where it's consistent with a pharmacopoeia monograph. So you've defined the manufacturing process, the key critical tests and specifications. We want to be assured of the consistency of the manufacturing to these parameters. So you should provide a summary of results on tests on at least 2 consecutive batches of the finished product. And these batches should be representative of the manufacturing process. You should provide stability data on 3 consecutive production batches, stability studies under proposed storage conditions, and on reconstituted products, if applicable, under normal and recommended handling conditions. The stability profile of product is important, not only for the regulatory authorities to ensure product quality for the shelf life of the product, but also for the manufacturer for both live products and inactivated products. Whereas the end of shelf life titer can be defined by the efficacy dose response studies, the minimum batch release titer will be defined by the stability profile at the recommended storage temperature. For products less than 1 year, we recommend monthly for the first 3 months, and then every 3-month intervals after that. For products greater than 1 year, for the first year, every 3 to 4 months, the second year, every 6 months, and then subsequent years after that, just testing annually. Stability studies should be properly designed, conducted and reported in a manner consistent with good laboratory practice. A summary of the proposed shelf life, storage conditions, and justification for the proposed shelf life should be provided. Real-time studies should be carried out on the final finished product in the marketed container. It should include descriptions of each of the tests, and these should be the physical, the chemical, the biological, or microbiological aspects linked to the safety of the product. Liquid products should be stored inverted during the stability study to demonstrate the integrity of the seal. And stability studies may include additional tests on top of those listed in the expiry specifications. For example, vacuum testing, which gives assurance of the container material. A test for sterility and safety should be conducted at the final time point in the stability protocol. For multivalent vaccines, each active ingredient should be tested. The results of stability testing should be consistent with and confirm the minimum release titer and end of shelf life specifications for the product. If the product contains a diluent or solvent, stability studies for these are required as well. In use stability, the continued integrity of the product in multi-dose containers after the first opening is an important quality issue. The repeat opening and closing of a multi-dose container may pose a risk to its contents with regards to microbiological contamination, proliferation, and/or physiochemical degradation once the closure system has been breached. A minimum of 2 batches, at least pilot scale, should be subject to testing. One of the batches should be within an appropriate period of the expiry date of the product. The other batch can be tested at any time point of manufacture. Appropriate data required to demonstrate that the vaccine retains an acceptable quality profile and remains efficacious throughout the in-use shelf life. You should identify the relevant parameters to efficacy, potency, and safety. Microbial safety of the vaccine should be demonstrated over the proposed in-use period. At a minimum, the efficacy of the preservative should be demonstrated. And you should take into consideration real-world usage when designing these in-use stability trials. Take into consideration external influences such as sunlight and heat. So that is the things we look at on the chemistry and manufacturing side. When we come to efficacy and target animal safety, I'll start with efficacy. Efficacy of the vaccine means the induction of the immunity to provide protection against a specific disease. The nature, degree, onset, and duration of immunity are the main parameters of the protection. All claims for the efficacy of the vaccine, including the duration of protection and the administration schedule, should be fully supported by data from specific laboratory trials and field studies. Efficacy data should be generated in Australia for the registration of all veterinary vaccines intended for use in food-producing animal species, unless strong scientific argument is provided that overseas data is applicable to Australian climatic conditions, genetic stocks, and farm management practices. General requirements for these studies: they should be conducted in the most sensitive class or member of the target species. The dosage used should be that of the end of shelf life or lower. For live vaccines, the vaccine agent should be at the highest attenuated passage level that will be present in the vaccine. And these should be from products that have gone through or will be representative of the manufacturing process. Clearly defined in the study protocols the parameters to be measured in efficacy trials and give justification in relation to the indications and specific claims for the vaccine. Conversely, you should give justification for not measuring parameters that are usually related to the disease of concern. There's 3 types of parameters. Clinical parameters, these are your, for example, mortality, morbidity, lesions, weight gains. You've got indicators of immune response, for example, serological responses. So for an indicator to be acceptable as a correlation of vaccine efficacy, you should show that a sufficient qualitative and quantitative correlation exists between the indicator measured and the claim protection in the target species. And then you've got indicators of infection, viremia, organism shedding, transmission. You should compare a group of vaccinated animals with unvaccinated controls, negative controls. For modified live vaccines whose vaccine agents may potentially spread, it's appropriate to separate vaccinates from controls. In such cases, separate housing of the 2 groups would be justified. Each trial should involve a sufficient number of animals to give statistical analysis. A well-defined challenge model should be used that as far as possible mimics the natural conditions for infection, particularly the amount of the challenge organism and the route of administration. The trial results should supply clear evidence that the vaccinated animals and control animals have been exposed to the pathogen of interest. Observations of signs of disease are rarely sufficient by themselves, and wherever possible, clinical records should be supported by laboratory tests. The cause of any deaths or unexpected signs of disease related to the parameters being measured should be determined. And in the case of live vaccines, wherever possible, the isolated field strain should be differentiated from the vaccine strain. Ideally, the trial should be double-blinded and placebo-controlled. So the types of studies required, the establishment of a minimum protective dose and vaccination schedule can be undertaken as a dose response study or as a study to confirm that the chosen dose and vaccination schedule are efficacious in the target species. You should justify the method for determining the minimum protective dose. And from the study, the end of shelf life titer can be established. The influence of maternal or pre-existing antibodies. Pre-existing antibodies against the vaccine agent may be maternally derived or be due to infection or vaccination. If the specific claims for the vaccines are related to efficacy in the presence of antibodies, trial protocols should include vaccination of animals with those maternal antibodies or antibody titers at the age of vaccination that normally occurs in the field. Onset of immunity looks studies on how long after administration of the vaccine before the vaccination provides protection against challenge. Duration of protection is the longest interval between the administration of a vaccine to target animals and the loss of protection against challenge. The level of protection against challenge should be consistent with the label claim for the entire nominated duration of protection. The response to revaccination is best demonstrated by challenge trials at suitable times between the end of the schedule and the end of the claimed period of protection. Field trials, unless justified, results from laboratory studies should be supplemented with supportive data from field trial studies. Trials should align with good clinical practice principles. Wherever possible, field trials should be carried out on a number of sites to ensure that the evaluation covers representative animals, houses, husbandry practices, and the environment geographical differences. The vaccine used should be, as mentioned previously, that at the end of shelf life titer or below, and animals should be exposed to natural infections. Safety tests are designed to demonstrate the potential risk from the product that may occur under the proposed conditions of use in animals. The potential risk versus potential benefits for the use of the product should be evaluated. So safety trials should be conducted in the most sensitive class or members of the target species. And the dosage to be used should be the recommended dose at the highest maximum release titer or potency for the vaccine. For live vaccines, the vaccination agent should be at the lowest attenuated passage level that will be present in the vaccine. Again, all these should have been produced under the manufacturing system that will be used. So studies required. Each trial should involve a sufficient number of animals in each group to enable relevant statistical analysis to be undertaken. There should be a negative control group in each trial. For finfish, the BP gives an idea of numbers, and in general, that's 50 fish per group are used unless otherwise justified. For parenteral vaccines, most post-mortem examinations should include investigation of the occurrence of effects such as pigmentation and adhesions. And the types of restudies required are a single dose effect. A single dose of the vaccine shall be administered to at the recommended dose volume and by the recommended route to the animal of each species and category, which is its intended, including animals of the minimum age and administration. The animals are observed and examined at least daily for signs of local and systemic reactions. They should be observed for at least 14 days after administration, and the vaccine complies with the test if no animals show abnormal or local or systemic reactions or signs of disease or dies from causes attributed to the vaccine. Again, from the monograph, For fish vaccines administered by immersion, the bath the fish for twice the recommended time, using a bath at twice the recommended concentration. Repeat administration studies. This test it's not generally relevant in situations where the product is only administered once in a lifetime. But if required, you should determine the timing of the repeated dose. But the interval between the repeat administration should not be any longer than the recommended on the label. Overdose. An overdose safety assessment of inactivated vaccines is not usually relevant unless you're using a new adjuvant or scheduled excipients. For inactivated vaccines, if an overdose study is required, normally 2 times is sufficient, and for live vaccines, the requirement is 10 times. You should monitor for signs of systemic or local site reactions for at least 14 days. If significant lesions are still present, observations should continue daily until lesions have subsided to an insignificant level. Abnormalities detected should be thoroughly investigated, and performance monitoring should be performed, including appetite and general disposition at a daily rate. Reproductive performance is required to assess the impact on future breeding animals. Examination of immunological function, where the vaccine is known to affect or could be expected to adversely affect the immune response of the animals vaccinated or its progeny. Suitable tests on the immunological function should be carried out. There are some special requirements for live vaccines. You should provide studies investigating the spread of the vaccine strain to non-vaccinated and non-target animal species. This will also form part of an environmental assessment, unless you can provide acceptable arguments to show that this cannot happen. Dissemination studies, investigating the secretion of the vaccine strain as well as the biodistribution of the organism and its dissemination in the body's predilection sites for replication as well as the injection sites. Reversion to virulence. This test is carried out using the master seed lot. At the time of inoculation, the animal in all groups are of an age suitable for recovery of the strain. Serial passages are carried out in targeted animals using 5 groups of animals. The microorganism recovered is used as the inoculum for the next passage. The observations include all relevant parameters typical for the disease that could indicate an increase in the virulence. And these results should be compared back to the clinical signs and other relevant parameters from the original 1-dose administration safety test. The probability of recombination of genomic reassortment with field or other strains should be considered. So that wraps up the safety and efficacy. But before I jump to bacteriophages, please be aware that there are other areas to be aware of. Data may be required to address human health, environment, residues and trade concerns. For example, if the vaccine contains a GMO, an environmental assessment will be required. And if, as mentioned earlier, using novel adjuvants, a residues package may be required. So I'll jump and change ship and talk bacteriophages. So there's a lot of research out there about bacteriophages at the moment, particularly with AMR going around as well. So some of the uses proposed include uses biocontrols, direct decontamination of live feed and eggs, phage-coated biofilters for reducing pathogen loads, and as use as feed additive and veterinary medicines, coating on commercial feed, powder pellets, etc. The APVMA doesn't have any specific guidance on bacteriophage at the moment. However, the European Pharmacopoeia has a general text for phage therapy medicinal products. It's a general chapter due to the immaturity of the field, and this is as it's been constantly developed. It provides a framework of requirements in general, but also some more specific details to be taken in consideration. Covers naturally occurring preparations as well as genetically modified phages, single phages, and multiphage preparations. So a lot of the requirements are similar to what I've just spoken about with the cell banking system. So the bacterial cell bank, on top of what I spoke about earlier with the vaccines, you also need to take into consideration such as things like phage sensitivity and absence of detrimental phages. The phage seed lot, you'll have identification, microbial purity, the phage purity. You'll look at the absence of intrinsic phage contaminants. Potency is normally detected by a plaque assay or other suitable method. The purified harvest, which is the equivalent of the antigen production process, which I spoke about in the vaccines, you'll look at things like identification, potency, microbiological examinations, residual reagents, so you'll have tests for residues of reagents used during production and posing safety concerns, and also tests for host cell impurities and contaminants, things such as endotoxin, exotoxins, host cell proteins, host cell DNA, and need to demonstrate that they're absent or within an established specification. The final product should include tests for appearance, identification, potency, microbiological quality, sterility, pyrogenicity. If it's a solid, things like water content. If it's liquid, pH. There will hopefully be a new chapter in the European Pharmacopoeia soon. It'll be a general chapter on bacteriophage potency determination, aiming at standardisation of potency testing of single phage preparations, and providing guidance for potency testing of multi-component mixtures of phages. It was out earlier this year for public consultation, which closed at the end of June. And then if you're unsure about data requirements, please contact the APVMA and ask for advice. You can go through our general inquiries, which can give you general guidance, things on how do I submit an application, etc. If you're after more tailored guidance, we've got the PAA advice, and we can provide information on types of assessment requirements, modules, fees, timeframes, types of supporting data that's required, types of trials needed to generate data. If you have trial designs and you're wanting us to check to see their feasibility, we can do those sort of reviews as well. It's also, PAA is also required if you plan to submit an international assessment. And when you do come through for a subsequent application, you can get rebates on the cost of the PAA. We also have the technical assessments, which we call item 25, and it involves the APVMA assessing data which may be used to support a future application. So you may have all your chemistry data, but you're still doing your, say, your duration of immunity studies, and you're wanting to make sure your chemistry data is going to meet requirements. We can assess that. If everything is found fine, you will resubmit the data you submitted there, plus the assessment report we provide, and we can waive that that module since it's already been assessed. Data may so data can be submitted to us to look at if there's any relevance to the safety, efficacy, and trade criteria. And I'll stop there. I'm sorry for the dry topic, but I hope that helps understand the data requirements that are required around these sorts of products. These flow through to the new technologies just as much as the older technologies.
Thanks so much, Nathan. I think I'm going to have to go back to the recording because, as you say, there's a lot of detail in there, but it would be good to have this recording available for everyone as a reference for those different areas. And I guess, yeah, my key takeaway is that although these new and emerging technologies are coming through, the same key criteria apply in terms of when you assess them for, be that a registration or a permit. Yes, there might be some slight differences. You talked about the live vaccines needing 10 times dose for a safety test, where other more traditional vaccines, it's the 2 times dose.
Yep.
So I guess thinking about how aquaculture and these more smaller, minor industries might be coming across these types of technologies, in a lot of cases, they might be developed by industry through research projects at universities. In terms of getting them from those really research style trials out to the real world, what's the ability to use things like research permits and minor use permits whilst some of that data is being collated? Because it could take a long time to meet all of those requirements. For a minor use type sector, are there opportunities to have a more staged approach?
Yeah, so the research permits aren't a backdoor to being able to get these into industry quicker. They're there to design to allow you to generate that data to support a application, whether that's for registration or permit. So on a commercial size scale, it's not so much an option, Jasmine. The best thing I can do is suggest, if you're a researcher or a company talking to researchers and that, is, as I mentioned early on, we require these things to be presented to us. I worked in a lab, and the quality assurance systems that go through those things aren't to the same scale we require to be assured of the safety of a product going out and being used commercially. So again, just plan it well, speak to manufacturers. If you've got that basic data showing really good promise, speak to your manufacturers earlier, speak to your crows or industry. I really went through the data requirements there because it's good for people to understand what is required, because if something comes to us or the manufacturer is not talking about particular, inactivation, kinetics, all that, you need to be aware that they are critical components that we are going to require. So having that plan up front and making sure you're ticking all those data requirements we require is the quickest way to get something through.
Yeah, and hopefully that has been really useful to a lot of people online today, just understanding those different components, particularly of the chemistry and manufacture, which are, they're not straightforward. So, all right, well, thanks everyone for persisting. We've gone a bit over time, but that's because there's been so many great questions. And yeah, look, if there's anything else that's come up through this webinar or subsequently when you're reviewing it again, please get in touch with me, because I'm happy to follow up with specific questions to Nathan or others at the APVMA if there's anything specific. I'll just pop up my email address here, so vet@seafoodindustryaustralia.com.au. If there's anything that you want to follow up on, please reach out to me. This FRDC project is all about trying to facilitate these sort of conversations and make sure that people have the tools and knowledge they need to be able to use and access AgVet products. So yeah, massive thank you, Nathan. Really, really appreciate your time that's gone into preparing this and being here today.
No, thank you for listening and coming along.
All right. So yeah, if anyone online has a burning topic that they'd like to see in our next webinar, we'll have another one sometime early next year. So let me know what topics you're interested in, and we can track down some people to talk about those. All right. Thanks, everyone. Have a good rest of your afternoon.